Hello everyone, apologies for the delay. A very warm welcome to everyone joining us for today's Select Science webinar entitled High Throughput Screening of Affimer Proteins for Use as Affinity Ligands Using a Label-Free Technology. My name is Frankie McDonald and I will be moderating today's presentation, stepping in for SPRi. I am delighted to introduce today's speakers. Jeff Platt, Senior Application Scientist at Avacta Life Sciences, and Shiraz Friedman, Product Manager at HORIBA Scientific. In this webinar, you will learn about the properties of Affimer proteins as next-generation scaffolds, with the potential to be used as novel biotherapeutics. Plus, hear about a label-free screening tool to assess Affimer binding and interaction with target proteins of interest. Just before I hand over to Jeff, I would like to mention a couple of things. Please feel free to ask any questions for the Q&A session at any time during the webinar by clicking on the Ask a Question button at the bottom of your screen. You can view the presentation in full screen by clicking on the tab at the bottom right of the media window player, and you can download related documents in the Resources tab at the bottom of your screen. The on-demand link will also be provided a few days after the webinar. Without further delay, I'd like to hand over to our speakers, and I would like to thank them for presenting to us today. Please go ahead, Jeff.
I'd like to use the time today to describe Avacta Life Sciences technology, which are known as the Affimer reagents, and in particular I'd like to describe their potential use as affinity ligands for protein purification, and also like to describe some of the high throughput screening techniques that we've used, particularly label-free technology to screen for these Affimer binders. So what are Affimers? Affimers are a protein scaffold technology, and which we typically use as affinity reagents. And they can be thought of most simply as an alternative to antibodies. Biophysically speaking, however, they're very different to antibodies. They're much smaller, they're small single-domain proteins. Usually the Affimer of the scaffold plus the binding loops has a molecular weight between 12 to 14 kilodaltons. The scaffold structure itself is derived from the highly conserved cystatin family protein fold, and this imbues the protein with a great deal of stability. Highly specific interactions to targets are generated via interactions through binding loops, insertion of random peptide sequences into binding loops 2 and 4 that are highlighted there. And importantly, the Affimer proteins, neither in the scaffold nor in these randomized binding loops, contain either cysteines or post-translational modifications, which I'll describe the importance of this later on. So what are the advantages of Affimers? Many of these advantages stem from the simplicity of the molecule itself. The first obvious advantage is the ease of manufacture. We produce Affimer proteins in simple prokaryotic systems, which means we can get robust production with high yields. The fact that there's no post-translational modifications in these proteins, as I mentioned earlier, means that we can get very low batch-to-batch variability. Again, the fact we use very basic molecular biology methods to create these proteins means that we can engineer them in a simple fashion. This can be via fusions to other functional proteins such as GFP or inclusion of peptide tags, or in fact generation of multimers. Now these can be either homomultimers, can take advantage of avidity effects to increase binding of Affimer proteins to a target, or we can generate multi-specific molecules where we string Affimers for different targets together. And this has found particular utility in a therapeutic context, although I won't have time really to go into details about that today. Another nice feature of the Affimers is, as I mentioned before, the Affimers, either the protein or the scaffold or the loops, neither of which contain cysteines. This means we can use basic molecular biology to add cysteines in a site-specific fashion, and this allows us to use the unique thiol chemistry of the cysteine side chain to either site-specifically attach labels or to immobilize the Affimers on the surface in an optimized fashion. Another key feature of the Affimers is their high stability. We see that the protein scaffold itself has a really high stability, high thermal stability. They have an unfolding temperature above 100 degrees. Addition of the randomized peptide loops can decrease this somewhat, although we still see Affimers typically have TM values above 80 degrees. And we also see that we can incubate the Affimer proteins for extended periods of time in relatively high concentrations of organic solvents and at a range of pH values. And the Affimers retain their function and their structure. So that's a little bit about Affimers, but how do we go about obtaining an Affimer for a particular target? So we receive a target from a customer, and the first thing we usually do is biotinylate the target so we can immobilize it on the surface, so we can do an in vitro screen. And we actually do a phage display, 3 rounds of phage display against our Affimer library, which has a high diversity. And the fact that we do an in vitro selection means that we can design complex selections, for instance, to drive affinity. And we can do this by having very long wash steps, which means that we select for Affimers with particularly slow off rates on the target, which means that they have high affinity. We can also select for high specificity by doing deselection stages against other off-target proteins that might cross-react with Affimers, or we can remove Affimers that show cross-reactivity at this stage. And we can also, because it's an in vitro process, we can do the screening, the selection in different background matrices. So, for instance, we can study an Affimer in a serum background so that we know that in the final application the Affimer will work in the environment that it's been selected in. At the end of phage display, the Affimers are cloned into an expression vector. And actually at this stage, in a typical project, we pick 192 colonies. This means that all the techniques we use downstream of this process have to be capable of screening in a high throughput fashion. And this is borne out by our typical primary screen, which is a bead-based flow cytometry assay. And this has a nice feature in that we can test the Affimers that we've selected against the target they've been selected to bind against, but also we can check to see whether they're cross-reactive against other proteins, for instance, and we can do this up to 30 target proteins. The validation is the final stage of our process, and this depends upon the application, so it can actually be real use of the Affimer, the final Affimers we selected in ELISA or a pull-down assay, for instance. And importantly, the whole process is very rapid. It only takes approximately 12 weeks from receipt of a target from a customer to handing over Affimer clones that bind to that target. So that's a little bit how we obtain Affimers. We've seen there's some real key features for these Affimer proteins, one of which is the fact that, as I mentioned before, we can reformat them in many different ways, but also we see they have a really high specificity. So we've been able to obtain Affimers that can distinguish between proteins that have single amino acid changes, between proteins with different post-translational modifications, and also that can Affimers that can recognise specific conformers of a protein out of a pool of different conformers. So this means that there's been a wide range of potential applications for this Affimer protein technology. And there's actually been a nice review within the last 6 months published in eLife that actually goes into a bit more detail about a range of these applications. However, I'll briefly summarise a few. We've seen they've been useful for pull-down assays, for high-resolution imaging, even as crystallization chaperones. But today I'd really like to focus on a couple of applications. One which I'll describe at the end is a capture reagent in ELISA assays, but mostly I'd like to focus on the use of Affimer proteins as affinity ligands in affinity purification resins. So the aims of this particular project was to demonstrate the ability of an Affimer-based affinity resin to reproducibly bind and elute a target under standard bioprocessing conditions. So these are kind of conditions where, under production, we've obtained a target protein that's in a complex background, perhaps a cell lysate or a serum. We can pass it down a column containing a resin that's been conjugated to Affimers, typically at neutral pH. The Affimer resin will bind the target and will wash away any contaminants, and then usually change conditions. This is typically to low pH, and then specifically elute the target. And the aim was to produce a data pack for the Affimer-based resins that establish a high degree of specificity for the target, even in these complex backgrounds, that we can produce these resins that can be reproducibly used again and again, and that will be robust to harsh cleaning conditions that are often required in bioprocessing. We also wanted to demonstrate that the binding capacity of these columns could approach those columns that have been available commercially and been developed over a long period of time. And also we wanted to demonstrate one of the key features of Affimers, which we believe is the real flexible formatting options, and we wanted to show that this could provide avenues to improving the characteristics of these Affimer-based affinity resins. So, for these particular projects, it would be impractical actually to screen all the Affimers we selected from phase display on an affinity column format. We'd have to do a lot of chemistry attaching Affimers typically to agarose-based beads that are used in these resins, and it would take a long time to do the experiments, so it just wouldn't be practical. And so in this case we use a proxy during our primary screening stage of SPR imaging, and in fact we use HORIBA's XL-Plex instrument, and this has lots of real advantages for us. Firstly, certainly compared to an affinity column format, it requires a tiny amount of sample. And also, the surface chemistry of the chip mimics that of the affinity resin. And if you look at the gold chip in the bottom corner there, you can see some of our Affimer proteins printed in an array on this chip surface. The fact that we can print on this surface our array of different Affimer clones means that in a single experiment we can screen many Affimer clones simultaneously. Firstly, we can screen for affinity by doing a titration of the target protein. We can screen for specificity by testing cross-reactivity against other off-target proteins. We can test the elution conditions, because it's important for this kind of application, not that just that the Affimer proteins can bind their target, but also they can elute them as well. We can also test on the chip that the Affimer proteins are robust to the harsh cleaning conditions required in this kind of application, and also we can test the effects of different formats to see if we can improve upon the characteristics by altering the protein. So the way we've developed the use of the HORIBA XL-Plex for this kind of application is we print typically 192 samples simultaneously on a single chip, and the diagram at the bottom, the image at the bottom left there, just shows a photographic image of the array. The fact that we can print so much on the surface in one go is obviously brilliant for time efficiency. We can obtain a lot of data within a day or two. We can get direct comparability between samples at the same time in the same experiment. Because often we can print so many spots, we can often print multiple spots of the same sample, meaning that we can check for reproducibility and gain error values for each sample. And obviously another thing that's important to us is actually the relatively low cost of these consumables for a high throughput SPR data that we get, because a single chip can obtain so much data. The way we've developed the use of these SPR imaging chips in our lab is to print the Affimers onto epoxy modified gold chips, so using a contact printer. And again, there's advantage of the fact we can use multiple spots for each chip. Means that we can vary the print conditions to try and optimize printing within the same experiment. And in fact, we often use pH to drive specific conjugation chemistry, because there's a couple of different ways that you can imagine a protein might immobilize an epoxy surface. The first one can involve attachment via interaction with primary amines, and these are typically found in lysines. Now there can be up 12 to 15 lysines in an Affimer protein, so it means we can have multiple orientations and multiple attachments. But more typically what we do is actually we use basic molecular biology to insert a single cysteine into the Affimer proteins, and this allows us to orient the Affimers in a single optimized fashion via selecting the right pH conditions to drive this. Once we have the Affimers on the surface, as we see at the start of this trace here, we can then inject across our target. And actually the image in the bottom right there shows actually the SPRi Response lighting up the chip as we inject target. You can see binding across the chip there. And for each individual spot we get a trace that looks like this, so we see kinetics for each spot. So we can see association, increased binding as the target comes in, and then we switch back to bulk buffer, and gradually the target washes away and we see dissociation. So we gain a lot of information very rapidly. So now I'd like to show some real data from a real customer project that's been anonymised. The aim here was to produce a stable affinity resin that could bind a target. However, the target's present in a complex mixture that also includes some closely related off-target homologs. And in fact, it's important to say that the Affimers were selected against this target during phage display without deselection against these closely related target homologs. So it's likely that in this cohort of Affimers that have reached this stage of the project, there could be some that show cross-reactivity not just for our target, but also for these off-target homologs. And by this stage of the project, actually we had 20 Affimers that were measured by HORIBA's XL-plex. And this is some of the data that we obtained. As you can see, when we inject on the target, so if you look for Affimer 1, we can see in green there's a clear response upon injection of target. It shows nice binding kinetics. However, injection of the off-target homologs 1 and 2 in blue and red, there's no response at all, which shows that Affimer 1 has a great specificity, so this is a nice Affimer to take forward. Affimer 2 shows something similar, very high specificity, whereas Affimer 3, whilst it clearly binds to the target, it also displays reactivity with the other off-target homologs. And at this stage of the project, we would take out this Affimer because specificity is really key to this kind of application. And actually, after this study, we saw that the 5 Affimers that displayed specific binding to the target. We also did some orthogonal studies by pull-down assay, and they agreed perfectly with this SPRi data and showed that the same 5 clones showed this high degree of specificity. So it was a nice validation of the technique. Actually, on the next slide now, the workflow involved here actually doing a titration of target to gain affinity data, but I don't really have time to go into that here, because what I'd like to show you something is an interesting study that we did that's particular for these kind of affinity chromatography studies. Now, it's important for protein affinity ligands to be stable to caustic cleaning conditions that are used every few cycles in bioprocessing to clean up the columns, and this involves relatively high concentrations of sodium hydroxide being incubated in the column. And we wanted to test that our Affimers weren't sensitive to these kind of conditions. So we tested that again on this same chip. So what we did here was we took a single concentration of target, we injected it across the chip, and the responses are shown in black. We then exposed the chip to sodium hydroxide, 100 millimolar sodium hydroxide, for various amounts of time, up to a total of 76 minutes contact time, and we measured the response at different time points. And if we look at some of the data in Affimer 1, for instance, we can see that initially after 3 injections of sodium hydroxide, there's actually a slight drop in the response. However, even once we double this time, over 6 injections, we could see that really it stabilised, which led us to believe that this Affimer does seem to be quite robust actually to this kind of treatment. Certainly if you compare it to Affimer 2, which instantly upon injection of sodium hydroxide at the early injections, it shows a big drop in its response, which stays low. And it indicates that this Affimer is probably too sensitive for sodium hydroxide to take any further forward. So we didn't do this. We didn't take this forward. And actually, after this whole process, we could see that there were 2 candidates selected that displayed the high specificity that I described in the previous slide, and this high robustness to sodium hydroxide that I mentioned here. We also saw from the affinity measurements that they had KD values in the ballpark of 100 nanomolar, which is approximately the right range that is required for an affinity resin. And so actually these clones were then taken forward and used to prepare affinity resins. So that was a nice use for us of this SPRi instrument. I also wanted to show actually, for this particular project, really how much data we obtained, all within about a day or so. Each of these traces actually is done in triplicates. There's actually more data there than might appear. So we could really obtain a lot of data. And one thing we also wanted to check was benchmark this data against other measurements. So we actually took some of this data and also data from a range of projects where we've got Affimers from a range of with a range of affinities to different targets, and we sent them out to an independent researcher, and we sent them blind to a CRO, and they used BioCore to measure the absolute KD values for this range of Affimer clones. And we compared it to the data we obtained on the HORIBA XL-plex. And what we saw was that there was very good correlation between the absolute values we obtained in the XL-plex with those in BioCore. And this gave us real confidence that not only can we get affinity and specificity data from the XL-plex, which helped with screening, we could also have great faith in the hard numbers that we're also generating. So I'd like to continue now talking about a different project. This is, again, it's an Affinity Chromatography project. However, this is a model system that was used in an internal study just to exemplify the use of Affimers in this kind of application. So we used an Affimer that specifically bound human IgG. And again, we did similar methods as I described earlier. We used the SPRi imaging alongside ELISA to test for specificity. So in this case, you can see we tested against human IgG, and we saw nice binding, and we also saw no binding against other species we tested, so dog, rat, and mouse IgG. So it shows it's specific for human IgG. We also could gain affinity data on the SPRi instrument, again, which was in the right ballpark for an affinity resin. And we also saw that generally we regenerate the chip surface on the SPR at low pH, and these are the same kind of conditions that would be used to elute protein from a column, and this Affimer could withstand those kind of conditions. So the fact that we could see the right kind of specificity and affinity, as well as the ability to withstand low pH, make this Affimer an ideal candidate for an affinity chromatography resin. So that's what we've prepared. We took this Affimer reagent and we prepared it with a single cysteine, in this case at the C-terminus, and we conjugated it to agarose that have been modified with iodoacetyl chemistry, which interacts specifically with the cysteine. We often use epoxy chemistry as well with agarose, which obviously ties in very nicely with the SPRi chip chemistry. We then take this modified agarose containing the Affimers and pack them into a column, as I've shown there. And we do a range of experiments that look a bit like this, where we take a target, in this case pure human IgG in PBS, and we load it onto the column. And you can see in this particular case, we loaded on the target at 0 mils. There's nothing comes off. Effectively, the Affimer column has captured everything there. We then, in the dotted box you can see there, we switch to an elution buffer, which in this case is glycine containing 50 millimolar sodium chloride at pH 3. And you can see during this elution phase we see this nice sharp peak appear, which is pure target. So we could see we could bind and elute specifically our target from this column, and we saw that we could do this reproducibly. So now we wanted to test a number of the key features of the column, because we see that the Affimers can work in this kind of context, which is nice to see. So the first thing we wanted to test, again, was the specificity on whilst the Affimers conjugated to a column. And we did this in a couple of ways. The first way was we tested in a complex matrix. So we took Ig depleted human serum and we loaded that across our column. And the trace is shown here. So it's shown in blue. It's actually quite difficult to see. But what you can see is at the start after the injection, there's a very large peak where most of the components of the serum pass through the column without binding at all, and so there's no cross-reactivity. Once we switch to elution buffer, again shown in this dotted box, we can see for the blue trace there's just a flat line indicating that nothing's bound. So it's really clear that the column doesn't bind any components of the Ig depleted serum. We then took this same serum and spiked in our target human IgG and reloaded it. And this is now shown in the red trace. Again, immediately after injection there's a large peak as most of the components of the serum pass through. However, now during the elution phase you can see there's a sharp peak, a sharp red peak. When we took the fractions corresponding to this peak and ran them on a gel SDS page, we could see that the eluted peaks corresponded perfectly with the reference lane for human IgG. So it shows we've purified our target from this very complex background mixture. We also tested the species specificity, and we did this in a different way. We took pure target proteins from IgGs from different species and injected them across the column. And we measured the area underneath the peaks and saw what percentage of the area is in the eluted fractions. What we saw for human IgG is that the column basically captured all the protein injected and eluted it specifically. For the other species we saw little or no binding, and this agreed nicely with the SPR imaging data that we obtained on the XLplex. So we can see that this resin had very nice specificity, and in fact we sent this resin to a third-party company for a PK stability study of human IgG drug conjugates in a mouse serum background, and they did this assay alongside using our resin alongside a well-known commercially available resin specific for human IgG, and they saw results that were effectively the same, indicating that for this kind of PK R&D study, our Affimer resins with very little development can achieve similar responses to those for highly developed commercially available products, which is really nice result. So another key feature of the Affimer resins we wanted to test was their robustness and their reproducibility. So the way we took this is we tested this was we took an Affimer column and we injected our pure target. We washed it with PBS, we then eluted at low pH as before, we re-equilibrated in PBS and then injected more target. And we repeated this over and over again, actually in this case 81 times. And also within these 81 injections we included 18 cleaning in place cycles. So these are to mimic the really harsh cleaning conditions that are required in bioprocessing, and in this case involved us exposing the column to 100 millimolar sodium hydroxide for 10 minutes. And what you can see, if you look at this graph here, the percentage area of eluted peak, you can see that there's very little change actually over the course of these 81 injections, and it shows that both the Affimers and actually the chemistry used to attach the Affimers are robust to these kind of cleaning conditions and also to the high amount of cycles they've gone through. Now, importantly, we also pulled together all the fractions corresponding to eluted target, so it contained pure target. We pulled them together, we concentrated them up 240-fold, and then we ran them on SDS page and Western blot. And on the right-hand side here, there's just an example of the Western blot with 2 reference lanes for pure Affimer. And you can see the lane where I've pulled together the eluted peaks. We can't detect any Affimer, which shows that there's little or no leaching of the Affimer from the column, even under these strong, quite harsh cleaning conditions used. So it shows that again that these columns appear to be robust under these difficult conditions. Another feature we're interested in measuring is the potential capacity of these Affimer columns. Now, we did this in a couple of ways. I should say at this point that the columns that we're using were test columns that were actually very small. They've only got 0.6 mil bed volume. So they're not very large columns, but we tested them in a couple of different fashions. The first way was to do a simple titration, and that's shown here. If you actually look at the blue squares on this graph, it's simply just injected increasing concentrations of target, which is human IgG, and we can see that we get most of it back. Even when we injected on 12 mg onto this very small column, we were getting over 11 mg back, so we were pleased with that, and it shows that we had potential to bind a great deal of protein. However, another feature we're keen to demonstrate is the fact that the Affimer proteins can be formatted in different ways to improve their characteristics. For instance, we could put cysteines at a range of sites. But another thing we wanted to test was the effect of linker length. So we actually genetically manipulated the Affimers using basic molecular biology to include 3 extra residues of glycine between the Affimer and the C-terminal cysteine. So effectively increasing the linker length between the agarose beads and the Affimer. And we wanted to see what effect that would have. And we studied this via dynamic binding studies. In these studies, you take column and you run across pure target. And eventually you see saturation of the column and breakthrough. And you can see this in the top graph. You can see this breakthrough, and then there's an increase in the absorbance measured. We then switch back to wash buffer, wash this away, and finally switch to low pH elution buffer. And you can see we get nice sharp elution peaks at the end. The traces in red correspond to the Affimers with this increased linker length. And what we can see is, for this elution peak, we actually managed to bind and elute more target protein when we had this increased linker than with the shorter linker length. We also see, if we look at the trace below, that the actual the volume required before saturation increases when we have this linker length, so this breakthrough volume. So it seems that by this very simple change to the formatting of the Affimer, we could actually improve the characteristics. And interestingly, we also saw on the XL-plex, when we tried this Affimer with a longer linker length, we also saw an increase in the Rmax, so perhaps that was a hint already that we could show improvements with this version of the Affimer. So I'd just like to finish this part of the talk here, just to remind you that for our model human IgG system, demonstrated that we could provide reproducible binding and elution profiles under conditions typically used in bioprocessing. We could see high specificity for target even in the complex matrix conditions I described. But we saw high robustness even in the harsh caustic column cleaning processes. We think we've got the potential to bind high concentrations of target. And I've shown that we can easily format to optimize characteristics, and there's a lot more development we can do here. There's a lot of things we can try. From the point of view of the HORIBA XL-plex, the SPRi provided us with not only with really valuable primary screening information, including high throughput affinity and kinetics data, but also provided things that are important for this Affinity Chromatography project, which is information about specificity, the robustness, and formatting information. So it was a really valuable technique in this instance. So I'd just like to finish off with just very quickly going through another application of Affimers before I finish. We've done an interesting project recently. We've obtained anti-idiotypic Affimers. So these are Affimers that combine specifically to the CDR loops of therapeutic mAb. So they combine specifically to a particular therapeutic mAb. And we've been demonstrating the applicability of these in PK assays because the availability of the capture reagents for therapeutic antibody PK assays can be rate limiting in product development. The capture reagents, often you have to use the native target. They're not always reliably available and they can be expensive, as well as which they can be prone to specificity issues and matrix problems. Development of anti-idiotypic antibodies to replace them can also be a difficult process, so we believe that Affimers can provide a fast, reliable solution for this therapeutic antibody capture. And so the proof of principle we underwent was to develop an anti-trastuzumab binder in collaboration with Covance. So the aim would be, in this particular diagram here, to replace the natural ligand for trastuzumab, which is HER2, with an Affimer protein. So again, we went through our selection process. We selected against biotinylated trastuzumab in the phage display. We deselected, in this case, against a therapeutic antibody cocktail, and we also used an off-rate selection step to select for Affimers with high affinity, because that's really key to this kind of application. And eventually this was whittled down to 22 clones, and this is actually where the HORIBA XL-Plex came in again. Here's just some example data here where we've immobilized Affimers on the surface, and we've injected across a range of different mAbs. You can see when trastuzumab is injected, the Affimers show a range of responses showing binding to this mAb, whereas the other well-known therapeutic mAbs, there's no response at all, indicating high specificity for our target. The next thing we're interested in was obtaining affinity data for these Affimers to the target. Now, one of the downsides, I guess, of having been able to collect a lot of data in one go is being able to sift through this data and work out which data is really worth analysing. And obviously we had 22 different Affimer clones here. And one of the nice features that HORIBA's put in the software is this cloud function, which enables rapid ranking. So if you look at the top left, there's a range of response curves there for our Affimers against a single concentration of target. And we can select 2 points, one during the association response and one during the dissociation response. And we can plot the absolute association versus the percentage dissociation on this graph below. And this is done in triplicate, and you can see straight away there's a couple of obvious candidates shown in green and orange that show a high initial association response, but also very slow dissociation. And because affinity is really key here, these would be good Affimers to really look closely at their kinetics. And that's what we did. There's an example here of clone B3 that shows single-digit nanomolar binding. We took our lead clones and we tested them in an ELISA in-house. We simply physiosorbed the Affimers to a maxisorb plate, came in with a biotinylated target, and then detected using streptavidin HRP. And what we could see is that the ELISA confirmed that the Affimers bind to trastuzumab in a comparable fashion to that of HER2, both in terms of IC50s and limit of detection. We didn't see any matrix effects either when stood in the presence of serum, and actually we saw that they were comparable to the anti-trastuzumab antibodies that have been used in ELISA recently. We actually sent a number of these Affimers to Covance, and they instigated a PK assay according to European Medicines Agency guidelines, and they saw that the accuracy and precisions were exactly what would be expected, and they also saw some improvements. For instance, the working range we saw a broader working range than that obtained with the current antibody-based PK method, which is really nice. So that was just a very brief summary, and I'd just like to finish with that, which is to say that we actually obtained binders in that project within 13 weeks, and we saw that the Affimer proteins performed comparably to HER2 as a capture reagent. We also saw they competed with HER2 in a competition ELISA format for trastuzumab. I haven't had time to show the data there. Just wanted to say the validation of the reagents by Covance demonstrated that Affimer binders can be qualified as critical reagents in a clinical PK assay. and they actually had some benefits in terms of the low matrix effect, the broader dynamic range. We saw no batch inconsistency when we tested over four batches. Of course, we know that we can supply them easily. The idea would be to put an immobilized Affimer on the surface here. So with that, I'll finish. I'll just like to remind you that some of the advantages of Affimer proteins, they're quick to develop. We can control the specificity due to the fact we're using an in vitro selection method. The biophysical properties of the protein themselves mean they're small, robust and stable. The simple production means we can get consistent batch reproducibility. And as I've mentioned a few times, we can use various formatting options, and the fact we can introduce cysteines, specific sites, means we can easily functionalize these proteins and orient them in any way that you can think of. So I'd just like to finish there and say thank you very much for your kind attention. Thank you.
Brilliant. Thank you, Jeff, for an extremely informative and enjoyable presentation. Now we're going to move on to the final part of today's webinar, the question and answer session. So, question number 1. How many target proteins would be required to screen your Affimer library for binding?
How many target proteins, or how much target protein? Sorry.
How much target protein?
Yeah. Sorry. Yeah. Well, actually, we need very small amounts of protein. Typically, we'd say between 50 and 100 micrograms of protein. That will allow us to do the phase display selection, also the primary screen, and even the validation step, depending on what application it's required for.
Perfect. Thank you, Jeff. Next question. Can you produce Affimer-based affinity resins that bind and elute other protein targets?
Yeah, that's a good question. Obviously, in the presentation, I focused on a specific application, which was purification of human IgG. And the reason why we use this as a target really is because there's a lot of information there about currently about purification chromatography columns that purify IgG. So we could benchmark our data against these systems really nicely. And also the target is also very cheap and practical to use because we use quite a lot of it. But I can say we've got lots of customer projects where we've raised Affimers against different kinds of targets and been able to purify them. Internally, we've got information where we show nice purification of a GST from complex mixtures. And we've also got very interesting binder that binds actually to fragments of IgG. So we have an Affimer column that can bind specifically to the Fab domains of IgGs, and it binds to the Fab domains and to the intact IgGs, but it doesn't bind to free light chain or the Fc domain, and this differentiates it from a currently available resin, Protein L resin, which binds to free light chain, so therefore it cannot differentiate between free light chain and intact Fab, whereas we have an Affimer resin that so far appears to do that, and we're still developing that resin. So that's quite an interesting binder that we have that we've been using in column chromatography.
Okay, great. So question number 3. One of the key features of Affimers that you mentioned was high specificity. Can you give examples and specific details of any systems where this has been shown?
Well, we have quite a few. A lot of them, fortunately, are customer projects, so it's difficult to go into detail about those. However, there's a couple of nice examples. Indeed, there was a couple of publications last year where an academic group have shown differentiation between recognition. I've got Affimers that can recognise ubiquitin dimers that are linked via different lysine side chains, and so they can recognise these Affimers that are these ubiquitins that are linked via different lysines into dimers via isopeptide bonds. And it's been impossible to raise antibodies against these, specifically against these dimers. So that's a really nice example, and there's a recent publication in Molecular Cell and Nature Structural Molecular Biology describe that. We also have nice examples where we can specifically bind to toxin B from C. difficile and differentiate from toxin A, and this has proved difficult in the past with other systems. So that's another nice example.
Fantastic. Right, question number 4. How much Affimer is typically conjugated to the agarose resin?
In the test resins that we've been doing, we typically, using the iodoacetyl chemistry on these agarose beads, we typically obtain 10 mg of Affimer conjugated per mil of resin. Now we're also testing other chemistries, on different resins actually. So there's potential that we could improve on that. There's a lot of room certainly for development with Affimers in terms of how we could conjugate them to agarose beads, for instance. So I'm sure we could develop this further.
Okay, great. So moving on, how much Affimer protein is used in the Exelplex SPRi assay?
Well, I guess Shiraz might want to answer some of this, in a broader sense. But I'd say for Affimers we use tiny amounts. So we typically print 5 micromolar of the Affimer, which is about 60 micrograms per mil, and we use usually 350 micron diameter pins. And we know that it only deposits nanoliter amounts, so we only use nanograms per spot. And we reckon about 1 microgram could do more than 6 spots for sure. So we use hardly any Affimer protein in the Exelplex assay. So it's one of the real nice features.
Perfect. So next question is, which type of negative control did you use?
Oh, well, I can say for Affimers, in this case, it's actually quite nice for us. We don't always subtract a negative control, but if we do, we often have print spots that just contain Affimer scaffolds, so they don't contain any binding loops. So that's quite a nice negative control background to see there's non-specific binding of the target as it's injected over the surface. So it's kind of a perfect negative control really for us.
Katie, can I go back to the question, the one before this one?
Yes, please do.
Yeah, because I think if I well understood, the question is the number of different Affimers we can immobilize on the SPR biochip. We can, in the case of Avacta, we immobilized 384. One of the experience we did with Avacta, but we are able to immobilize even more than 400 different Affimers in the same biochip.
Great, thanks Shiraz. So the next question that we've had in is, can you employ nanodisk or detergent or lipoprotein-based membrane proteins as targets?
That's a good question. I've not been involved in projects that have done this myself, but I know we have worked with other companies where we've done work like this. I believe with GPCRs. So it's definitely possible. I should say, we can raise Affimers not just to proteins, but pretty much we can try it with any kind of target really that we can biotinylate that can go into our selection process.
Okay, brilliant. So we've got time for one more question. So the next question is, how do you immobilize the Affimers on the surface of the biochip?
Well, I can say, I can describe it there. We have been using the epoxy. I should say there's a range. I'm sure Shiraz can fill you in with more detail, but from HORIBA there's a range of chips available with different surface chemistries. And we have been using as a kind of a go-to chemistry is the epoxy surface. And we've seen that's been working really well for Affimers. However, we also use malamide surface chemistry, which enables us to really specifically interact with the cysteines that we typically insert into the Affimers. I don't know whether Shiraz has more details for different kinds of surfaces.
Exactly. If the question is related to the surface chemistry, so in the case of the Affimer, we are using specific surface chemistry, but there is a different kind of functionalization of the biochip, which can be used for different kind of molecule, protein, peptide, or antibody, or different kind of molecule. If the question is related to the way to immobilize the Affimers on the surface, there is a different way to immobilize molecule on the surface, but in the case of Avacta, they used the immobilization of the Affimer through direct contact spotting. It's a pin which takes the solution and go in contact with the surface, and the solution is immobilized. The molecule is immobilized in this way.
Okay, perfect. So that's unfortunately all we've got time for today. Thank you so much to Jeff and Shiraz for today's informative discussion and presentation. And thank you to everyone joining us online and for your really interesting questions. I hope that everyone's found this a really worthwhile session. If we didn't manage to answer your questions, then feel free to email me at frankie.macdonald@selectscience.net. I will follow up your questions personally. If you would like to listen again to today's webinar, or even invite a friend to listen, it will be available to watch on demand in a few days' time. Goodbye, and thank you once again for joining us.
Goodbye.
Thanks. Bye.